Hello everyone, welcome to this session about staining. So, what exactly is staining? Staining is nothing but a procedure to stain. Now, what is this stain? Stain is a dye. Okay, so putting a dye on any media is known as staining. Okay, staining is the process of putting dye or any stain. Okay, so why is it important? What is it? So, for example, if this is any dish and we want to view, like we have smeared some organisms. Okay, these are organisms that we have smeared and we want to see it clearly, then we need to put some stain so as to visualize it better. Okay, to visualize it better. Now when we put stain, we can see the shape, arrangement, even the morphology, and it also gives a rough idea about the type of organism. So we can select the antibiotic treatment accordingly. Okay, now I'll make it even clearer to you. If this is any dish, okay, if this is any dish, if this is any dish and these are the organisms, are you able to identify these normally? No. But now if I put any stain, will you be able to identify the organisms? Yes. So this is the use of stain. Okay, so for better visualization of the shape, arrangement, morphology of the organisms, and the type of organism can also be identified, which helps us to select the antibiotic therapy. Okay, so this is all about the stain. Now let us discuss the types of stains. What are the types of stains? There can be simple stains. That means that simple stain basically means that you took one dish, there were some organisms, you stained it, it took the color. So if it takes the color, then it is simple or direct stain. Now you put the organism here, suppose it has capsule. So what happened is everything else except the organism and the capsule, everything else except this capsule got stained. That is, this capsule did not get stained. So other things got stained but the capsule did not get stained. This way you can identify the organism here. So this is negative stain or indirect stain. You know, a very good example of it would be Cryptococcus neoformans, which is a fungi. Now what is it? It is a fungus. Number one, what does it cause? Meningitis. Okay, mainly meningitis, but other lesions can also be caused, other injuries like other diseases can also be caused. Mainly it causes meningitis. So as I said earlier, all the organisms which go into the chest or to the brain are capsulated. So Neisseria meningitidis, Cryptococcus neoformans is also one of them, along with Streptococcus pyogenes, Streptococcus pneumoniae, and Haemophilus influenzae. Clear? So Cryptococcus neoformans is capsulated organism which can be identified by indirect staining. Okay, indirect staining means that the organism or its capsule would not be stained, but everything else would get stained. But in simple staining, the organism itself gets stained. Okay, so these are about the types of staining: simple or like direct or indirect. Okay, there are various methods. And Gram stain, Ziehl-Neelsen stains are very famous. We'll discuss about it now. So, what is Gram stain? Gram stain basically occurs in four steps. We have four steps for it. Number one, we put one primary stain. Okay, so primary stain. For example, if this is one dish, we put crystal violet blue. So if you put crystal violet blue, everything will turn. For example, if this is crystal violet blue, everything will turn into this color. Second step would be to put iodine. We also call it mordanting. So what is mordanting is basically when we, for example, if this is an organism, okay, this is an organism, okay, it has cell wall, and cell wall is made up of peptidoglycan. So when we put any dye, the dye penetrates this cell wall and it lies in the cell wall layer. This crystal violet basically is in the cell wall, right? Now if we give iodine, then this iodine forms bond with the dye. So if this is dye and this is iodine, they form a bond, so that when we decolorize it, this bond as a whole can come out. You got my point? So if these are organisms, this is the cell wall, this is the cell wall. So dye is in the cell wall. Dye is in the cell wall. Now we give iodine. Iodine forms bond with the dye. Okay, so when we apply any decolorizer is any substance which would remove the dye. Decolorizer removes dye. Okay, so decolorizer is there to remove dye, but the role of iodine is that iodine forms bond with dye, because of which removal is easier. Without iodine, this decolorizer would not be able to remove the dye. Clear? Okay. So first step is to put the primary stain or crystal violet blue, which is taken up by the cell wall. Second is putting an iodine. The step is called mordanting, in which the iodine forms a strong bond with the dye. Third step would be decolorizing. Okay, and the fourth and the ultimate step would be to put a counter stain. Now I'll make it very clear. Firstly, we put one crystal violet blue dye. Okay, secondly, we put iodine, which formed bond. First step was this, second was iodine. In third step, what we did was, there are organisms, okay, which took this stain. What happened is that when we applied decolorizer, dye plus iodine complex would get removed, right? Would get removed normally, okay, and all the surrounding structures would also get removed, would also get decolorized. This thing, second thing, happens. Okay, but this dye and iodine complex getting removed in case of gram positive organisms, And in case of gram-negative organism, it is very different. So in gram-negative organism, you might be knowing peptidoglycan layer is very thin. Since it is very thin, this iodine and dye complex gets completely removed. Okay, but in case of positive organism, peptidoglycan layer is very thick. So dye plus iodine complex does not get removed. Clear? So what I meant is, now first step was we put this dye. It was taken by gram positive and gram negative both organisms. It was taken by both organisms. But in case of gram positive, what happened was that the surrounding structures, they got they got decolorized, but this cells, their cell wall, did not get decolorized. These organisms did not get decolorized. Organism did not get decolorized. But in case of gram positive, in case of gram negative organisms, what happened is that we put this stain. These are the organisms. Suppose these are the organisms. Suppose, okay, these organisms, firstly they took the stain, but when we applied decolorization, all the stain was gone. Clear? So till step 3, this is all that happened. Now comes step 4. We are applying counter stain. Counter stain is basically carbonyl fusion, which is red in color. Now, in case of gram positive organisms, they had retained this crystal violet blue. Crystal violet blue is retained. But in case of gram negative, the surrounding plus the cells had no color. So what would the obvious thing be? The obvious thing would be that these organisms, when we put carbonyl fusion, which is red in color, okay, carbonyl fusion is red in color, these organisms would be like this only, right? So what you see is, in case of gram positive, there are purple cells against red background. When we put this counter stain, there are purple cells against red background. In case of gram negative, there are red cells against red background. Clear? So this is what we obtained, and from there we classified. So from gram stain, which has 4 steps, okay, gram stain which has 4 steps: primary staining, mordanting, secondary staining, decolorizing, and finally counter staining. There are 4 steps. In case of gram positive and gram negative, when we applied primary staining, both took purple color. When we applied iodine, to both. Then when we did decolorizing, gram positive retained the purple color. So the cells still had a purple color, but in case of gram negative, it was no color. No color. Because it got decolorized. Why did it get decolorized? Because the cell wall is thin in case of gram negative. Then final step, when we applied red color, it was red cells against red background. But in case of gram positive, since they retained the purple color, it was purple cells against red background. Clear? I hope I could make it clear. Now let us talk about Ziehl-Neelsen staining. This Gram stain, okay, it is aqueous based. That is, it is water based. Now, can water based stain penetrate any cell which is very, very, very thick in lipid? Can it penetrate? No. Okay, so this is the story of Mycobacterium tuberculosis and other organisms which have very thick cell wall. Okay, thick cell wall as in lipid rich. Thick cell wall, that is lipid thick. Okay, lipid rich. Now, this Mycobacterium tuberculosis basically has thick mycolic acid which does not allow the penetration of aqueous based solutions or aqueous based stains. Okay, so what to do in case of MTB? So in case of MTB which has mycolic acid rich layer, there are 3 steps that we follow. Number 1, primary staining. So what we do is we apply carbonyl fission, which is, again, you know the color, right? It is red in color. And as I told you, it is very thick. So just applying stain will not work. So with stain, we give heat. Why heat? So as to allow the penetration of this stain. So as to allow the penetration of carbonyl fission, we apply heat. Okay. So heat in a way acts as more denting. Okay. So number 1 is primary staining with carbonyl fission, which we do along with heat. Okay. Now second step is obviously decolorizing. Now the thing is, this mycolic acid layer is so thick. Okay, this layer is so thick that it resists decolorization by acid. So here in case of decolorization, we try to give acid, and you know acid is very strong, but even acid failed to decolorize this layer, decolorize this carbonyl fission layer. Okay, so what we had done was we gave carbonyl fission stain, the cell took it up because we had given heat along with it. Now we are trying to decolorize it by giving acid, but acid fails to do it. That means the cell is resisting the acid. That is why we called it acid fast stain or acid fast organisms. Okay, why do we call them acid fast organisms? Because they resist. Fasting in a way is resisting. So in a way they resist the acid decolorization. Okay, so but will they resist all types of, like, all concentrations of acid? No. Okay, so they will resist up to 20% H2SO4, beyond which they will get decolorized. So up to 20% H2SO4 they resist. Okay, so till now what happened was, this is petri dish, this is the organism, for example Mycobacterium tuberculosis, this is the carbonyl fission. Okay, just simplifying it, it does not actually look like this. Okay, in fact if I draw it properly, it is a bacilli, it is not even a cocci, it is bacilli. So bacilli, and it has thick carbonyl fission layer. So anyhow we managed to give carbonyl fission. Number 1 step was carbonyl fission, right? So number 1 step is carbonyl fission. So anyhow we managed to give carbonyl fission. That means the cell has now become red, right? The cell now becomes red, right? Okay, and what about the surrounding? Yes, obviously it also gets red. First step done. Now let me take the same thing as an example. Second step is that we applied decolorizer, which in case of this is H2SO4, 20%. Mind you, beyond 20% it will get decolorized, but up till 20% it won't get decolorized. So what that means is that the cell will retain the stain, that is, it will retain carbonyl fusion. What about the surrounding? Yes, the surrounding loses the stain. Okay, so what happens is surrounding lost the stain. Okay, this is the second step. Now third step. In third step, what we do is we give Loeffler's methylene blue. When we give Loeffler's methylene blue, will the surrounding take it? Yes, the surrounding takes it. What about this Microbacterium tuberculosis? No. Why? Because something which could resist acid, will it get stained by it that easily? No, it won't get stained. We are not giving heat. And MTB are already stained with carbonyl fission. Since the Microbacterium tuberculosis did not get decolorized earlier, it won't get stained by another stain. But the surrounding was already decolorized, so it will get stained by crystal violet blue. Okay. So what do we see in case of Mycobacterium tuberculosis is that we see red cells against blue background. Clear? Now one more confusion. This is blue background, clear. Okay, this is blue background, okay, done. But so far I've learned that this Mycobacterium are beaded. You know what beaded are? Beaded is like this type of arrangement. Okay, why are they beaded? Why? Because my friend, this carbonyl fission, you know how we had given it? We had basically given heat with it so as to allow penetration of the stain. But will it get uniformly penetrated? No. This mycolic acid layer and the cell wall is so thick that the penetration is not uniform. So the penetration of carbonyl fission is not uniform, because of which the acid fast bacilli is not uniformly stained and it gives beaded appearance. Clear? So far what I've covered up is that in case of acid fast bacilli, we see red cells against blue background. Now I mentioned acid fast bacilli. So who all are acid fast bacilli? MTB for sure, which resists 20% H2SO4. Now are there others? Yes, there are modified Zait-Nielsen stain also. Modified Zait-Nielsen stain is that in Zait-Nielsen stain we use 20% H2SO4. In modified we'll use less concentration of H2SO4. Now why less? I already mentioned it. I already told you that if more than 20% decolorization is possible, okay, in case of H2SO4, so that means it will be H2SO4 as long as it is 20%. Okay, similarly there can be organisms which will get decolorized at 20% H2SO4 but might resist at lower concentration. Is it possible? Yes, it is possible. So these organisms are known as modified these organisms take up modified Zn stains, that is Zn stain having lower concentration. Okay, so these organisms include Mycobacterium leprae, which is given around 5% H2SO4. So at 5% H2SO4, it resists decolorization, that is, it is acid fast. But above 5%, it will get decolorized. So we use 5% H2SO4 just to view red leprae against blue background. Clear? Similarly, nocardia, actinomyces are other organisms which are given lower concentration of H2SO4, because at a higher concentration they will get decolorized. So at lower concentration they are given, so around 1%. Clear? So these are other acid fast bacilli. I hope I've made this clear. Now I will take you through one guide. Okay, read along with me. Read along with me session. So if you want to join, like, you can continue with this video, otherwise the session is done for today. So let's start the staining methods. Number one, what is staining? It is an auxiliary technique used in microscopy to enhance contrast. I already mentioned it, right? So to enhance contrast, we use staining. Okay, so staining helps in identification of any bacteria. It helps in starting the treatment. Okay, for example, gram positive bacteria are susceptible to penicillin G than gram negative bacteria. Now I'd like to explain why gram positive is more susceptible to penicillin G. So penicillin is beta lactam drug, which is cell wall synthesis inhibitor. Okay, and gram positive, they have, what do they have? They have thick peptidoglycan layer, right? Thick peptidoglycan layer. Also gram positive, I said there is NAM NAG arrangement. In NAM there is AGLA. This L and A are connected by transpeptide bond 1 2 3 4 5, and transpeptide enzyme is used. And I said penicillin inhibits this transpeptidase enzyme, right? So for penicillin to act, it will inhibit transpeptidase enzyme. When it inhibits transpeptidase enzyme, this process does not occur, this cross linking does not occur. Okay, but in case of gram negative organisms, they are directly cross linked, there is no transpeptide linkage, so there is no use of giving penicillin in gram negative. That is number one. But what is number two? Number two is that in gram negative cell wall, there is there is one inner membrane, outer membrane, and periplasmic space in peptidoglycan layer in between. In this periplasmic space, there are enzymes, beta lactamases. Now I mentioned just now that penicillin is a beta lactam drug. So penicillin beta lactamase is an enzyme which will break down beta lactam structure. So basically what I mean is gram negative bacteria have an enzyme called beta lactamase which can destroy the beta lactam structure of penicillin. Just so it basically inactivates the penicillin drug. Clear? Okay. So this is the reason why gram positive bacteria are more susceptible to penicillin G, and thus, like, if we can identify any organism as gram positive, how will we identify that? We'll identify as purple or blue cells against red background are gram positive. Blue against red, right? We just saw it in gram stain. So blue against red is positive organism, and positive organisms are susceptible to penicillin G, so we can start the treatment likewise. Clear? Now what is stain? Stain is any substance which adheres to cell, gives it a color. Okay, so why should we stain a bacteria? To observe it under the microscope, like better visual, I said, right, for visibility. Okay, to visualize it better. Okay. Now, types of staining. Simple staining can be direct, which is positive, crystal violet blue, methylene blue, carbon fusion that we just did. Okay. Negative staining, I mentioned already, Indian ink. Now, there are for better understanding, you can go through this slide. Not very important. Then I'd like to take you to gram staining. What is gram staining? 4 steps: primary staining, which we used crystal violet, then mordanting using iodine, decolorizing using we used one dye, like we used iodine, right? Iodine was used, and counter staining, which is done using carbon fission. This table is very important. Okay. Gram positive bacteria, purple, 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 and gram negative, purple, purple, colorless, and red. I I have made it earlier, so just refer to it again. Why some bacteria are gram positive, some are not? They are positive because they retain the primary dye complex. And negative because they do not retain it. Decolorizing agent used during the staining disrupts the membranous envelope, and the dye and iodine complex is washed out. Okay. Then why is Lugal's iodine used? Iodine is used if iodine is not added, crystal violet alone will not be extracted by organic solvent. That is why. Okay. Then what is Zerin staining? What are acid fast bacilli? These are for your own recapitulation. Just try to answer it in your own mind. What acid fast stains? Because of lipid mycolic acid in the cell wall, sulfuric acid cannot penetrate into the cell. That is why decolorization is prevented. So they are called acid fast. And the bacteria don't contain mycolic acid, they are easily decolorized by sulfuric acid, so they are not acid fast. Okay. The why some bacteria are acid fast, why some others are not? Because of mycolic acid, already you know. Why acid fast bacteria can't be stained by gram stain? That you know, because gram stain is aqueous. Okay, aqueous based stains such as gram stain cannot stain mycolic acid, which is lipid layer. Then a modified Zerin stain, you know that, right? 5% H2SO4 by lepri, actinomyces. Okay, nocardia, these are. Then acid fastness is the ability of the organism to resist Zerin stain. Okay. And acid fast bacilli are the organisms or the bacilli cocci, never cocci, okay? Never cocci. These are bacilli which are stained by Zerin stain, are known as acid fast bacilli. Mycobacterium tuberculosis, lepri, actinomyces are example. So, to summarize, gram positive organisms, okay, gram positive organisms appear as violet or purple against red background. Gram negative appear as red against red background. Then acid fast bacilli appear as red against blue background. That's it. Okay. And talking about the method of staining Zerin stain, we know primary by carbon fusion, decolorizing by H2SO4, and counter staining by low fluorescence methylene blue. I hope I've made it clear. Thank you for watching this session. Thank you.